Okay, hello to all of you. Let's discuss chapter number 10. So chapter number 10 is about photosynthesis, and we in the chapter number 9 we have already talked about cellular respiration, and we know that these two processes, they are related, and they are the products of cellular respiration, the byproducts of cellular respiration. They are used for the process of photosynthesis. So photosynthesis is a process in which solar energy is converted into chemical energy within the chloroplast. So photosynthesis is a process which is conducted by the photosynthetic organisms, which are not only plants, but there are algae, there are some bacteria. They can also undergo photosynthesis. And photosynthesis is a process which is actually nourishing the whole living world, because the main source of energy is coming from the sun, and this solar energy or light energy is converted into chemical energy, and that energy is stored in the form of plants. And so either we as humans, we either depend on plants directly, or we eat animals who have been fed on the plants. So the main source of energy is basically coming from the sun, and by the process of photosynthesis, the light energy is converted into the chemical energy. Now, what are autotrophs? Autotrophs are those organisms which can make their own food, which can basically take the sun's energy and convert it into chemical energy. So photosynthesis, as I said, is done by plants, by the algae, by cyanobacteria, purple sulfur bacteria. So it's not only by plants, it is also done by bacteria and the algae. Heterotrophs are those organisms which cannot make their own food, so they are dependent on other organisms. Like us, we are heterotrophs. We cannot make our own food. So we either eat plants or we eat animals that have been fed on the plants. Okay, so a little bit about the site of photosynthesis in plants. So basically, this is a cross section of a leaf. In the leaf, there are some cells called mesophyll cells. In the mesophyll cells, each mesophyll cell has around 20 to 30 chloroplasts, 30 to 40 chloroplasts. And in these leaves, there are some pores called stomata from which the exchange of gases can take place. And we have already talked about the structure of chloroplast. You can go back and review, but I can quickly go over quickly go and give you the review of the structure of the chloroplast. So basically, as we have already discussed, there are two layers, the two membranes, and there's the outer membrane, then there's the inner membrane, and inside there is these disc structures called thylakoids, and these thylakoids are stacked like a stack of a pancake, and that stack is called granum or grana. And here is a jelly kind of a material called stroma, and in the stroma is the DNA of the chloroplast as well as the ribosomes. And the chlorophyll, which is the main pigment involved in photosynthesis, is in the thylakoid membrane, and we're going to discuss very shortly details of the chlorophyll, the pigments. So photosynthesis is a process which involves some reactions, and the overall reaction of photosynthesis can be written as carbon dioxide plus water plus light energy converts into glucose and gives oxygen and water. So this is the overall equation for photosynthesis. 6 molecules of carbon dioxide plus 12 molecules of water plus light energy. The input of energy gives rise to glucose, 6 molecules of oxygen and 6 molecules of water. So it's basically the reverse of cellular respiration. So in this process, there is a release of oxygen. So the process of photosynthesis not only gives us glucose, it not only gives us the food, but it also gives oxygen. And the source of oxygen is by the splitting of water. The source of oxygen is by the splitting of water. So these are the reactants of photosynthesis. Carbon dioxide plus water, and they give these products which include glucose, water and oxygen. So the oxygen which is coming out as a product in the photosynthesis, its source is basically water. So photosynthesis, a very important thing to know about photosynthesis, it's an endergonic process. Okay, it's a process which um, involves the input of light energy. Okay, so cellular respiration is a exergonic process, it's a catabolic process. On the other hand, photosynthesis is a anabolic process and it is a endergonic process, and it needs the input of light energy. And photosynthesis is a redox process in which water is oxidized and carbon dioxide is reduced. So this is basically explaining photosynthesis, input of energy. So it's an endergonic process. Carbon dioxide plus water gives you glucose and oxygen. So carbon dioxide gets reduced and water gets oxidized. So photosynthesis involves two phases. One is called the light reactions, or there are two stages of photosynthesis: light reactions, or it's also called photo part because it involves sun's light energy, and second is called Kelvin cycle or synthesis phase, or it's also called dark phases. So these are the four main events which takes place in the light reactions. So light reactions is the part of photosynthesis which involves sunlight. So in this part, splitting of water takes place and it releases oxygen. And also in the light reactions, NAD plus. So we have already talked about in cellular respiration, the electron carrier is NAD plus. But in the photosynthesis, the electron carrier is NADP plus. Okay, there's one extra P from plants. So the electron acceptor NAD plus gets reduced to NADPH, and ADP is converted into ATP. So these are some of the important things which takes place in the light reactions. Water splits and gives oxygen. ADP gets converted into ATP, and NADP plus gets converted into NADPH. And the energy is converted into two forms. One is ATP, another is NADPH. And these two forms of energy, they are used in the second phase of photosynthesis, which is called the Kelvin cycle. And in this Kelvin cycle, the reduction of carbon dioxide takes place, and the source of energy in this phase is ATP and NADPH, and ATP and NADPH, they were produced in the light reactions. So this is showing you overall photosynthesis. So this is the first phase. So this light energy is taken by the pigments which are there in the thylakoid membrane. These are thylakoids, and in this, splitting of water takes place and there is release of oxygen, and there is also production of ATP and NADPH. And this is Kelvin cycle, in which carbon dioxide is fixed or carbon dioxide is reduced, and it involves the input of energy in the form of ATP and NADPH, and it results in this carbon dioxide is reduced and it forms the sugar. And the Kelvin cycle takes place in the stroma. Okay, so we will be basically in the photosynthesis, we are going to talk in detail about the light reactions, how ATP and NADPH are produced, and then we are going to talk about Kelvin cycle, that how sugar is produced in the Kelvin cycle. So before that, we need to know some of the terms or some of the basic information about the sunlight. We need to know about the electromagnetic spectrum of sun's light. We need to know about pigments, how the pigments are there in the plants. They are present in the form of photosystems. So first of all, we need to know about some of the basic terminology and basic concepts, and then we are going to move towards the process of photosynthesis, which involves the light reactions and the Kelvin cycle. So sun's light is in the form of the electromagnetic, light is basically electromagnetic energy, or it's also called electromagnetic radiation. And the electromagnetic energy, it travels in rhythmic waves. Okay. So the electromagnetic spectrum is the entire range of the electromagnetic energy or radiation. So it involves all different kinds of radiations, like, so this is the visible light. Okay, so this is the visible light from the sun's electromagnetic spectrum, which reaches the Earth's atmosphere. There are other radiation also in the sun spectrum, which involves ultraviolet radiation, infrared, microwaves, radio waves, X-rays, gamma rays, but it's only visible range which reaches the Earth's atmosphere. And in the visible range, you can see it ranges from 380 to 750 nanometers. So it's a visible light which is taken by the plants, and that light energy which is in the visible light range spectrum, that's the one which is taken by plants, and it is used for the process of photosynthesis. So pigments are substances that absorb visible light. Okay, so pigments, and there are different kinds of pigments. So this coming week you will be doing a lab on fermentation and photosynthesis, in which you will be working with pigments and all that. So you'll come to know more about the properties of the pigments. Different pigments, they absorb the radiation of different wavelengths, and some of the wavelengths which are not absorbed, they are going to be reflected or transmitted. So the plants look green because the chlorophyll, they reflect and transmit the green light. The leaves basically look green. So you can see here that this is the sun's light, the visible range, the visible part of the electromagnetic spectrum of sun's light, which is being taken by the pigments which are there in the thylakoid membranes. And basically the blue and the red part of the sun's visible range is absorbed, and the green part is basically transmitted. So that's why the leaves look green in color. And in the lab, you will see we use an instrument called spectrophotometer, and spectrophotometer we have already used in the enzyme lab. So it can basically tell us the pigment's ability to absorb various wavelengths. So this machine sends light through pigments and measures the fraction of light transmitted at each wavelength. And with the help of the spectrophotometer, we can make an absorption spectrum. And what is absorption spectrum? It's a graph plotting pigments light absorption versus the wavelength. So absorption spectrum is a graph which is made by plotting the pigments light absorption versus wavelength. And we are also going to make absorption spectrum in the lab. So this is basically showing that if this is your chlorophyll solution, and if you pass the green light through the chlorophyll solution, because green light is not going to be absorbed by the chlorophyll solution, but it is going to be transmitted. And when the transmitted light is detected by the galvanometer or the spectrophotometer, basically it's going to show very high transmittance. And if you pass blue light through the chlorophyll solution, if you pass the blue light through the chlorophyll solution, then this light is going to be absorbed by the chlorophyll solution, because this is the range of the wavelength which is absorbed by the chlorophyll. So in the spectrophotometer, you will get very low transmittance, because the blue light is not getting transmitted, but it is rather getting absorbed. Okay, there are three different kinds of pigments in the chloroplast. Chlorophyll A, which is the key light capturing pigment, this is the main pigment. Then chlorophyll B is the accessory pigment, and then there are carotenoids. They are also separate group of accessory pigments. So the absorption spectrum of chlorophyll A suggests violet, blue, and red light works best for the photosynthesis. And then we have another spectrum called action spectrum. So action spectrum basically tells us at which wavelength the photosynthetic organisms they can perform the best photosynthesis. So action spectrum profiles the relative effectiveness of different wavelengths of radiation in driving the process of photosynthesis. So this is showing you the absorption spectrum. So you can see this is chlorophyll A, And so this is the absorption spectrum for chlorophyll A, this is for the chlorophyll B, this is for carotenoids. So you can see the one peak here for chlorophyll A, this is one peak for one peak here for chlorophyll A, and this is the another peak here for chlorophyll A. So it absorbs the light best in this red region and in this blue region. This is the action spectrum. This is basically telling this is action spectrum, and the action spectrum basically tells the rate of photosynthesis with the at the different wavelengths. So it's telling that this is for I think this is for chlorophyll A. Yeah, so this is basically the rate of photosynthesis is being monitored by the production of oxygen, right? Because oxygen is a product of photosynthesis. So you can see here, one peak you see here, one peak you see here. So that is why it corresponds with the absorption of blue light and the red light here. And this is basically an experiment, very old experiment done by a scientist called Engelmann. So now we have various monitors which can actually monitor how much oxygen is being produced. Like this action spectrum is on the basis of the oxygen produced, and there are many these days oxygen monitors which can monitor how much oxygen is being produced. But this is an older experiment done by a scientist called Engelmann. So in this experiment, what he has done is he has taken this algae, which is a photosynthetic organism, and he is exposing this algae to different wavelengths of the light. And when this scientist is exposing this algae to the different wavelengths of light, and he's trying to study at which wavelength the photosynthesis is maximum, for that he's not monitoring the oxygen, but what he's doing instead, he's taking the aerobic bacteria, and what he's finding when he's exposing to this algae to the blue range of the visible light, he sees a lot of growth of the aerobic bacteria, and he also finds a lot of growth of aerobic bacteria when he exposes the algae to the red light. So the growth of aerobic bacteria is a measure of the production of oxygen, because oxygen is the product of photosynthesis. So aerobic bacteria needs oxygen. So if there is more oxygen, aerobic bacteria needs oxygen. If there is more oxygen produced because of photosynthesis, there will be more growth of the aerobic bacteria. So this is what I just explained is written here. Okay, so this is basically the action spectrum of photosynthesis, basically, not only for any specific pigment, and it's showing it's more broader. It's showing the action spectrum is more broader as compared to the absorption spectrum of these different pigments, and it is broader because there is not only one pigment, but there are mixture of the pigments. So the action spectrum photosynthesis is broader than the absorption spectrum of chlorophyll, and it is all because there are accessory pigments also, like accessory pigments such as chlorophyll B broadens the spectrum used for photosynthesis. And this is the structure of the chlorophyll. This is the general structure of a pigment. So it has the pigments, they have this ring called porphyrin ring, and this porphyrin ring is basically light absorbing. Basically this is the light absorbing head of the molecule. So this is the part of the pigment. This is the part of the pigment which absorbs the light, the sun's light. And this is the hydrophobic tail. This is the hydrophobic tail, and this part of the pigment, is actually embedded in the thylakoid membrane. And this is basically showing here is the methyl group for chlorophyll A, and there is instead of methyl group there will be this aldehyde group for the chlorophyll B. So the structure of chlorophyll A and B is basically the same, but only these are the differences in these groups, these functional groups. So basically, as we are discussing, that these pigments they absorb sun's light. So what happens to these pigments when they absorb sun's light? So when the pigments they absorb sunlight, okay, when they absorb sunlight, this is the photon from sun's light. the electrons in the pigment, in the chlorophyll molecule, they get excited. So the electrons in the chlorophyll molecule, they get excited, and when these electrons, they fall back, they release heat, as well as they release some photons, and that is what is the fluorescence. So which means that these pigments, when they get the sun's light, the electrons in the photos, the electrons in the pigments, they get excited. When they fall back, they release the energy in the form of heat as well as the light, and it has some color. So that's why this is called fluorescence. So in isolation, some pigments also emit light, and afterglow called fluorescence. So when these pigments, they emit light, it is of some color. So this process is called fluorescence. But this happens when the pigments are isolated. Okay. In the plants, when the photosynthesis takes place, in the plants, the chlorophyll, these pigments, they are in the thylakoid membrane. But this kind of a thing doesn't happen in the plants with the pigments. The reason is, these pigments, they are not isolated. Okay, these pigments, they are present in a system called photosystem. Okay, so this is the photosystem. So this is a thylakoid membrane. Okay, this is a thylakoid membrane, and these green dots you see, these are the pigments. Okay, and this is also a pigment. So these pigments, they are not present in an isolated system, but rather they are present in a system called photosystem. And what this photosystem is? This photosystem has two parts. Okay, it consists of a reaction center surrounded by light harvesting complexes. Okay, now the reaction center is an association of protein holding a special pair of chlorophyll A molecule and a primary electron acceptor. So this is basically the photosystem has two parts. This violet thing you see, this is a protein. This is a protein with these pigments on it. Okay, the pigments are present. They are embedded in the protein. We just saw the structure of the pigment. This is the light. This is the part which is going to absorb the light. This is the hydrophobic tail. This is the part of the pigment. This is the part of the pigment which is embedded in the thylakoid membrane. So this is a photosystem. This photosystem has this is a light harvesting complex. This is a protein with these. It's a protein with these pigments, and this is a reaction center. In the reaction center, this is a protein with these pigments, a special pair of chlorophyll A molecules, and this is the primary electron acceptor. Okay. So what happens in this process is, these pigments which are embedded here in the light harvesting complex, they absorb sun's light, and then they pass this sun's energy from one pigment to another pigment. So you can see this is all passing of energy taking place, and eventually this energy reaches this special molecule of chlorophyll A. This is a pair of chlorophyll A molecules here. The energy reaches here, and this energy makes the electrons get excited in this chlorophyll A molecule, and these electrons are eventually taken by the electron acceptor. These electrons are taken by the electron acceptor, and as a result of that, this chlorophyll molecule, it basically loses the electrons. So this gets oxidized, and this primary electron acceptor, it accepts the electrons, it takes the electrons, and then these electrons passes, then this electron acceptor passes the electrons to further more proteins, which we are going to discuss very shortly. So I hope everybody understands you guys understand the structure of a photosystem. So photosystem has two parts: light harvesting complex and reaction center. And there are, in the process of photosynthesis, there are two kinds of photosystems involved: photosystem 2 and photosystem 1. Photosystem 2 works first. Okay, so in the thylakoid membrane, you will see there are two photosystems there. One is photosystem 2, another is photosystem 1. Photosystem 2 is also called P680 because it absorbs the light at this wavelength. And the other photosystem is photosystem 1, and it is also called P700 because it absorbs the light at the wavelength of 700. So photosystem 2 comes first. Okay, it was discovered first, and photosystem 1 comes later. Okay, so I think I'm going to stop this video here, and I'm going to make a second video for explaining the linear and the cyclic flow of electrons.